welcome to this next video on OCR A-Level Biology. This is the last video on Module 2.3, Nucleic Acids. And in this video, we're going to be looking at protein synthesis. So to start off with, what we might do is actually discuss a little bit about RNA and some of the differences between DNA and RNA. So let's move on. So, oops, I need you. Okay, so let's move on to sort of different. So we ended the last video by talking about DNA replication. We discussed that DNA is a self-replicating molecule. Excuse me, and that the replication of DNA is semi-conservative. But now we're going to start looking at protein synthesis. And to do so, we're going to first look at RNA. Now we covered RNA in the first video on this module, but let's go through it once more time, in case you've forgotten. So, RNA is structurally different from DNA in a number of different ways. So first thing, RNA and DNA, they have different sugars. So RNA has ribose, has the sugar ribose. Remember, RNA stands for ribonucleic acid, but DNA stands for deoxyribonucleic acid. So therefore, DNA has deoxyribose. Another main difference is that RNA, the bases in RNA are slightly different to the bases in DNA. So RNA has uracil instead of thymine. So DNA has thymine, RNA has uracil. And another major difference is that the polynucleotide chain, the chain of nucleotides in RNA is generally shorter, is much shorter than in DNA. So the polynucleotide chain in RNA is much shorter than DNA, and RNA is usually single-stranded. Now, there are three forms of RNA which are going to be helpful in protein synthesis. So RNA comes in three forms in protein synthesis: mRNA, rRNA, and tRNA. mRNA stands for I'm going to write it down here. Stands for messenger RNA. rRNA stands for ribosomal RNA. tRNA stands for transfer RNA. So that's a little bit on RNA. So now we're going to look at, first of all, quickly before we go into protein synthesis, we must acknowledge that a little background. So proteins account for 75% of an organism's dry mass. They have many different roles within the body. So some proteins are structural, such as the cytoskeleton threads inside cells, or the proteins in the cell membrane. Others make up enzymes, hormones, and some may be used as antibodies or receptors or even channel proteins. So genes remain within the nucleus. Genes remain within DNA inside the nucleus. The proteins are made in the cytosol at the ribosomes. As the DNA cannot leave the nucleus because as DNA cannot leave the nucleus, the genes must be transcribed into a form of RNA, and RNA is able to stay outside the nucleus. So our mRNA is made from a section of or a gene of DNA, and this is done by the enzyme RNA polymerase, which we'll get to in a moment. And then once that happens, the mRNA, as you can see in this picture, leaves the nucleus and is then binds to a ribosome. This ensures that the coded instructions which are present in the mRNA are then translated into a sequence of amino acids and the protein is assembled in the correct manner. So I mentioned this before, so the genetic code is near universal because in almost all living organisms, we have 20 essential amino acids, but there's 64 different combinations of codons. So one codon is basically a triplet of RNA bases or DNA bases. So one triplet is called a codon, and you can have, and since there are 4 bases, you can have 64 different combinations or 64 different codons. But you have 20 amino acids. That means that one amino acid could have multiple codons. So, for instance, there's I think there's 6 different codons for leucine, which is impressive. So, even if a silent mutation occurs, it will not affect the amino acid and hence it will not affect the protein. The genetic code is also described as degenerate, is described as degenerate because, yes, so this is what I've just been saying now, describing that all amino acids except methionine and tryptophan, there is more than one base codon, and this may reduce the effects of point mutations. So what I was just describing there was degenerate. So the genetic code is universal, which means in all living organisms the same codon codes for the same amino acid. Degenerate, which means that one amino acid, except for methionine and tryptophan, there can be multiple base codons, which can reduce the effect of point mutations. And also the genetic code is non-overlapping, and it is read starting from a fixed point in groups of 3 bases. This means that if a frame shift or mutation occurs, so if a base is added or deleted and it causes a frame shift, then every base triplet after that, and hence every amino acid coded for, is suddenly changed, and that can affect the protein that is being built, and it may impact its function. So that is the nature of the genetic code. Okay, so now we're going to look at protein synthesis properly. So, protein synthesis can be divided into 2 major steps: transcription and translation. So transcription happens in the nucleus, and this is what you're looking at here. So transcription is when one section of DNA, a gene, unwinds and unzips. So the unwinding is unwinding the double helix shape, and then unzipping is the breaking of those hydrogen bonds between the complementary nucleotide bases. And then you have the enzyme RNA polymerase. So here it's very faint. It is very faint. Where is my pen? I don't know if you can see it, but this enzyme here, I don't know if you can see it, but see this thing I'm like waving around. This is the enzyme RNA polymerase. Excuse me. So RNA polymerase will bind to the DNA, to the gene, will bind to the start of the gene, and will catalyze the formation of temporary hydrogen bonds between the template strand. So this here is called the template strand. In order and will form a complementary mRNA molecule, which is complementary to the coding strand, which is this. So this is the mRNA that's been. Following that, the mRNA would then leave the nucleus via these nuclear pores. Whilst the DNA will remain within the nucleus. So remember, this is the enzyme mRNA polymerase. RNA polymerase catalyzes the formation of mRNA, catalyzes the formation of temporary hydrogen bonds between RNA nucleotides and their complementary unpaired DNA bases. So A bonds with T. Just to familiarize yourself here, A bonds with T, G bonds with C, C bonds with G. Now what bonds with A? It's U. U, because we're talking, this is for RNA, this is for the mRNA strand, and this is for the DNA. So A will bond with G, G will bond with C, C will bond with G, and then U, because we're talking about mRNA, will bond with A. Excuse me. Once the mRNA has been formed, there's usually a significant amount of mRNA editing that occurs. If you haven't already, you'll learn about this in year 2. But if you are doing year 2, then you should go and watch my cellular control video, which I have already made, which explains splicing and how mRNA is edited by the processes of splicing, whatever. But if you're in year 1, you don't need to worry about that until next year. Good for you. So this is mRNA. Once the mRNA has been edited, the pre-mRNA as we call it, once the pre-mRNA has been edited and forms mature mRNA, the mature mRNA will then leave the nucleus and travel to a ribosome. So a ribosome, I'm going to draw one, and will travel to a ribosome. And ribosomes are made up of 2 subunits. This one, so this is the large subunit of the ribosome. And then this is the small subunit of the ribosome. mRNA will come and then bind to so these subunits are actually found in the in the cell as separate subunits, and then they join together when and form the ribosome, which binds to the mRNA. So these ribosomes, a little bit about ribosomes, is that ribosomes, I'm going to ribosomes. These are made in the nucleolus, not the nucleus, the nucleolus. And they're formed of two subunits, as you can see in this diagram here, which I have drawn. And then these two subunits pass separately out of the nucleus through pores in the nuclear envelope, and then they come together to form the ribosome. So, so you have magnesium ions, so Mg2 plus ions, help formation of ribosome. So ribosomes, this is where the second RNA type in human cells comes in. Ribosomes are made out of rRNA. So this is mRNA. mRNA stands for messenger mRNA. rRNA stands for ribosomal RNA. So ribosomes are made of ribosomal RNA. But they're also contain smaller amounts of protein. Actually, no, they're not. They contain equal amounts of both rRNA and protein in roughly equal parts. There's also another type of RNA molecule which is going to be involved in this next step, which is translation, and that is tRNA. So oh yeah, I'm going to have to try and draw a hairpin type structure, because that's what tRNA molecules are. tRNA molecules have a weird hairpin shape. So let's see this bit here, and then let's do like this. So that's a rough shape of what a tRNA looks like. And then here you have the three bases. Let's do U, A, C. And then here you'll have A, U, G. I think you'll have up here. A, U, G. So this is a tRNA molecule, as you can see here. tRNA. Here, these are three bases make up the anticodon, which will bind to the codon on the mRNA. And these three bases here will actually bind to the amino acid. And for this particular tRNA, the amino acid here, I'll actually draw it in, is methionine. So methionine will bind to this particular tRNA. Okay, that aside, now let's go with protein synthesis. Here's a better drawing. So this is what the tRNA molecule will look like. So ribosomes catalyze the synthesis of polypeptides. And there's an excellent picture in page 93 of your textbook, which shows translation happening at a ribosome. It looks like it's an animation, but who knows, it might be a real life one as well. Who knows? Excuse me. Anyway, let's talk about what is happening here. So we're going to go through each of these step by step as shown in this picture. So step 1. So we have tRNA molecules. Bring amino acids in. Bring amino acids in. Actually, let's just do the general steps here first. Bring amino acids and find their place, and their anticodon binds to the corresponding codon on the mRNA. So tRNA molecules bring amino acids and their anticodon, depending on the anticodon, bind to the mRNA. So this is the I'm going to start labeling this diagram. So here we have our ribosome. This is our mRNA. These are our tRNA. Let's zoom in on this picture. Rather than me writing it all out, I think it's probably better just have you visualize it. So here we have, let's do an example with this mRNA. So here we have the ribosome. This is our mRNA. And we have these two are tRNA, which will bring their amino acid to the ribosome. So here we have the first codon. Let's do the first codon on this particular mRNA. AUG. So this is one codon. So this is one codon. AUG. The corresponding anticodon, which is this set of 3 bases here, this is the anticodon, is UAC. All tRNA molecules with the anticodon UAC will be carrying the amino acid methionine. So in it comes, you form temporary hydrogen bonds between the tRNA anticodon and the mRNA codon, and the amino acid is delivered. Now let's try the next codon on this mRNA. AUA. The corresponding anticodon will be UAU, which will have the amino acid tyrosine. So the tyrosine molecule, the tRNA with the tyrosine amino acid, will come in and also form temporary hydrogen bonds with its corresponding codon. As it does this, something called a peptide bond is formed. So this here is called the peptide bond. So that, as we mentioned in our biomolecules module, a peptide bond is what holds 2 amino acids together. So a peptide bond will be formed when 2 amino acids are joined together, and it releases, as we said, since it's a condensation reaction, it releases water in the process. Now, the next codon is after that, the ribosome would then move along and read the next codon, U, U, U. The corresponding anticodon would be A, 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 and that carries the amino acid phenylalanine. And then the one after that, CCG, that is lysine, GUU, valine, AUU, isoleucine, I think that is CCC, leucine, GAG, glutamine. But we'll get onto those in a minute. So the phenylalanine amino acid comes in and another peptide bond is formed. Also, while this is happening, the UAC tRNA will then be ejected from the ribosome, but it will leave behind its amino acid. But this tRNA molecule can now go pick up another methionine or another amino acid, or in this case, it's another methionine. Here you have peptide bonds. This will keep on going. So as you said here, this is the tRNA. And the cycle will keep on going. It will keep going until the protein building is finished, and then it will keep on going until the ribosome reaches a stop codon, which indicates that the protein building is finished. So these are your amino acids. And at the end of this process, you'll have a protein or a polypeptide chain, in fact. You have a series of amino acids, a chain of amino acids, which is a polypeptide chain. Eventually, this is the primary structure as we discussed in the last module. So we have a polypeptide chain, which is the primary structure. And eventually, this polypeptide chain will start to fold in on itself and start to go into the secondary, tertiary, and quaternary structure of proteins. And that is protein synthesis. So that is it for this video and for this entire module. So thank you all very much for watching. I feel that this might have been a bit short, a bit longer than the previous two videos. But this is a very complex and important process to learn. You need to be able to understand the details of how protein synthesis works. I'll leave links to two videos in the description below, which probably describe how this process works, how protein synthesis works a bit better than I have done. But anyways, thank you very much for watching. In the next video, we're going to be starting module 2.4, which is about enzymes. And boy, enzymes is a very big topic. So, yes, it's a very big topic, which it might take up, I don't know how many videos yet, but we'll see. So in the next video, we're going to be starting module 2.4, enzymes. But until then, thank you all very much for watching and hope you found this useful, and I'll see you in the next video. Goodbye.